All right, well, this is getting set up. It's a really great pleasure to introduce our next speaker, which is Dr. Kasthuri Venkateswaran. And if you can't easily pronounce the name like myself, you can just call him Venkat. Venkat has 2 doctoral degrees. He received his PhD in marine microbiology in India, as well as a doctorate in agriculture in Japan. He has more than 40 years of research experience in marine, food, and environmental microbiology. Venkat is a senior research scientist at NASA at the Jet Propulsion Laboratory, where he supports the biotechnology and planetary protection group. And his research portfolio is pretty diverse and also includes microbiology in clean room facilities, as well as the International Space Station. Now, before I will let Venkat kind of take it away here, I do want to share an interesting fact about him. So Venkat has been supporting students in India. Specifically, he's providing stipends for medical students. Now, there are some key requirements, and those are that the student is female, is raised by a single mom, and that she will do the same once she has a profession, just to keep this chain of helping hands going. So this is really a feed-forward mechanism. In honor of his mother's 100th birthday this year, he will graciously donate 1-year tuitions for scholarships for 100 students. And out of those, he's going to select the top 3 students and provide them with a full scholarship for their professional degree. So that doesn't only include the tuition, but that also includes the housing, etc. Boarding and lodging. So this is really terrific charity work. So I wanted to share that with you. And the charity name is Pichai, which is named after his mom. So with that, let's hear from Venkat. And unfortunately, he can't join us today because of the storm, the hurricane. He's going to be virtually. Take it away, Venkat.
Thank you very much for all. I hope that you are hearing me. Thank you for Tanya for the introduction, as well as Nikos for inviting me to present in JGI user meeting. Actually, I was so disappointed not being there because of the storm that we have gone through. All the flights are canceled. I think most of the other Southern Californian users are also not there. Anyway, so today I'm going to talk about the microbes in space, the challenges and the solutions for doing omics in space instead of bringing samples down. How we can do it in space. That's one biggest challenge that we are looking for. And when I say one of the biggest challenges, our samples are extremely low in biomass. Unlike you go to a kind of hot spring or some sort of the ground soil and other things. This is a femtogram level of DNA. So how we are going to work with the contaminations of the contamination of the reagents that we are using it for the next generation sequencing. So today I'm going to take you 2 examples. One is space station. The other one is the rover that's roving in Mars. So the samples that I am talking about are not from Mars. So these are from the surfaces of the spacecraft that is bound for Mars. But for the space station, we actually bring the samples down from space station to work on here. I think, I cannot stress the importance. These are really wild samples and apt for this particular session. So why the hell we need to do microbiology in NASA? Because it is an engineering institution. All of us can think about that way. But we are asking the biggest driving question for NASA is, are we alone? Are we Martian, for example? Are we coming from other planet? So that is the astrobiology question. So a lot of robotic missions are going there to look for life in other planets. That means better we clean our own spacecraft and the probes that are going and touching the samples and then looking for life in other planets. That is astrobiology. The other one is, we have to keep the other planet as is, as pristine as possible. We have to protect other planets that likelihood of having life. For example, think about Mars. So still we are not ruling out there is no life. But we already know that there is no life in Moon, our own moon. So that's why we are not taking extreme care when we go to the moon, but we have to take extreme care for the place where there might be likelihood of life, like Mars and the moons of the Jupiter, like Europa, and then Titan, Saturn's moon, Titan, etc. But we cannot keep quiet by not doing how these microbes reacts, because we are also simultaneously thinking about human habitation in other planet. So by doing so, we, for a sustainable presence in other planet, we need to think about those biological aspects of these microbes that might be able to withstand the space conditions, or whether they are going to be modifying itself to become a benign microbe, become a actually problematic microbe for human. So because of all these things, microbiology is happening in NASA. So today, I'm going to talk about the challenges in performing omics in space. As I already talked about the low biomass, reagent contamination, signal to noise ratio. Because we don't have that much time, and I'm not going to talk about the sample processing as well as instrumentation needed for automation. Because the people living in space, in the space habitat, are not too many people. You cannot take the elephant with you, but you can take a mouse along with you. In the sense, everything has to be small and other things. So, but that kind of instrumentation for automation, I don't have time today to talk about it, but with the JGA user, anybody have some, information so we can talk offline. So what are we learning from omics analysis that we have done so far? Genomics, proteomics, metabolomics of these space microbes and their functional property. And also we need to validate the predicted functions, whether that is useful for the Mother Earth, because we are all paying through the taxpayers' money to do the space science, and we need to get something back to our Earth. Actually, we came a long way to do the microbial monitoring of these spacecraft and associated surfaces. So, we started with, plate count, cultivable microbes that can grow in a given standard plate count is going to be, it started in 1960s, still we are using it. And this is specifically for spores, because spores are the likelihood survivor of the interplanetary transfer. But there are so many thermophiles and other radiation resistant microbes that are not spore formers, then that has to be cultured with some other way. As the previous person from Oak Ridge National Lab talked about it, if you know how to culture them, you will be able to culture the uncultured microbes. So not only this culture-based analysis, we are also looking into these, various molecules like ATP, LIL, and quantitative to enumerate the microbes, microbial burden, microbial biomass. And in cloning days, we cloned those 16S sequences in 1980s and 1990s. And then late 1990s and early 2000, we started using the DNA microarray. And then early 2010 and 2015 time period, we started using the 454. And then now we are using Illumina and Nanopore. We are not using PacBio mainly because the biomass is extremely low. It is not useful for getting the sequences. So this is an example for Mars 2020 rover that actually roving the Perseverance right now as we speak, and we were able to get the samples associated that around 250 plus samples collected during the 5 years time period, and then looking into what they are. This is actually a viable microbes. Viable microbes here, when I'm talking about that, trying to eliminate the dead DNA and then look for the microbes associated, DNA associated to the viable microbes. So roughly bacteria is dominant compared to the fungi. which is 1 to 2% of the fungi compared to the bacteria, archaea and viruses are extremely low. So more importantly, it is not what kind of microbial species out there. Main important is whether they are relevant for planetary protection, because we are containing the forward contamination when it comes with Mars, right? So the firmicutes, which are spores and radiation-resistant bacteria, and then the bacteria that can survive cold temperature, all those things are the one that I'm talking about here. Is roughly, when you compare, around 5 to 10% is what you are seeing of the total microbial population. So this is shotgun metagenome data right now. Next one is I'm taking you what is actually in space station. This is also based on viable metagenome sequencing using after sample processed with PMA to eliminate the dead microbes. So the IS microbe is unique to a particular subsystem, but also some microbial species are common. That is core microbiome consists. This is based on around 12 different missions that we were able to collect them. So the dominant microbes are mostly fungi, Penicillium or Aspergillus, because space station, human has to live, so you have a moisturized environment, otherwise human cannot tolerate. So when you have moisture, obviously, fungus will be there. But are they good? As we all know, microbes are not bad. We need microbes for healthy living. So these fungi are, as long as they are not, pathogenic, they're okay. But what I'm showing here are actually not pathogenic. But when it comes to bacteria, some of these things are really problematic, like Enterobacter cloacae, E. coli, Antiananatis, and Salmonella, and all this, and Staphylococcus aureus. These are BSL-2. In addition to the BSL-1, which is Staphylococcus epidermidis, which is a common, skin microbiome. But they are there, so this has to be appropriately handled. Otherwise, it will be problematic for long living in space. So when you compare with other microbiome studies, ISS microbiome represents a minimal core human model in which all the human subjects, because people are coming and going, it is a dynamically changing environment, So we have to be taken appropriate care about, how the microbes are moved along with the human, as well as the system, the cargo, as well as the animal that we are taking there to, to do for various experiments, right? So, and also it's a closed system. The air revitalizations are there. The air is getting pumped and then revitalized and putting back, because there is no outside activity you can have other than the confined external vehicular activity that spacewalk people are doing. But basically, what you have inside this closed system is airtight and hermetically sealed. Okay, having given all these things, Committee of Space for Research is coming up with some sort of thing. Hey, you guys are in DNA era. Why don't you actually use DNA-based technology instead of using only culture-based analysis, so that you will be able to get more specific microbial diversity data. So we had a multi-mission metagenomic technology development workshop for modernizing our planetary protection policy. I want to take a few minutes extra here to emphasize what NASA is doing, and here we need more input from the community, whether we are doing it right or not. So the workshop chair was Stefan, and Scott was, Scott Tai was a co-chair, and Thomas Torok also from another co-chair here. And we published this. It is not me. There is no NASA involvement here. It is purely non-NASA people, including some of the people from JGI and Lawrence Berkeley National Lab were involved in it. And we published this. They published this white paper. And that might be helpful for NASA to follow the requirements so that the future planetary protection policy can be modified. So these are the panelists that were there, from various other agencies like NIST, and obviously, JGI people represented Lawrence Livermore, Lawrence Berkeley, MIT, and other universities were there. So we have given some presentation what we were doing in NASA, and they evaluated and then come up with some recommendations. What is the motivation of this new method for bioburden assessment? Why don't we use the tried and dusted test that was doing since 1960s? As we know, culturable microorganisms are 1% or less, depending on which environment we're talking about. So in 1992 and 2006, Space Daddy recommended, actually, NASA has to adapt molecular assay. Let's give a careful thought about this language. These are all actual language given by them. Adapt. So that means it is not required. Then in 2019, they said required. So still, we are doing it on a R&D basis, research and development basis. It is not mandated. But in 2022 astrobiological data survey, the thing changed to should develop. So we are in an era that we have to get a technology, and NASA is coming up with the questions. It is not only what is in there is important. We want to know how much in there so that we can contain the contamination. So a hybrid technology that combines both culture-based and multiple next-generation sequencing methods as a standard methodology for spacecraft bioburden assessment needed. Okay, so NASA spent around 6 years worth of study on a research and basis, but as we know, for a federal government requirement changes, we need to validate this thing. So we wanted to see what are these technology out there that can be used by the federal agencies, academia, and industry. We don't want to reinvent the wheel. But one biggest thing is, can we do that for spacecraft associated surfaces, which is a low biomass? So that is the biggest issue. We need to get the rapid results. We need to get the quantitative results. At the same time, high information content results are needed for a low biomass. So workshop panel recommended a hybrid model, digital PCR, to get the semi-quantitative method, and shotgun metagenome to get a qualitative measurement of what are the things are there, because we need to know whether those microbes are indeed problematic for planetary protection and NASA missions or not. And biggest issue right now is bioinformatics, which will be helpful for linking this metagenomics and digital PCR, because based on the bioinformatics only, we will come to know some of the uncultivated microbes that can be used for digital PCR for quantitation. And we should not simply not doing culturomics because we have this omics, but we are also need to do this culturomics. That's what this workshop finding. So with this, what we are trying to do here is, get a model microbial community that is relevant for spacecraft, and then, dope it appropriately in relevant surfaces and collect. So this will tell us how much we are able to collect. And then appropriately we use the automated system, because technician A will be different from C and D. So we need to have some kind of automated system, and by putting the markup nucleic acids, we can extract the DNA and see how much we are losing here. Then we can back calculate, saying, hey, 99% you are losing here. That means if your requirements is 100 copies, so originally it might have 10,000 copies kind of thing. But that DNA will be used for absolute quantitation, and also the same DNA will be doing DNA library preparation and doing NGS library, and then get the metagenome-based analysis. And then this will be helpful, the bioinformatics pipeline will be helpful to select what are the microbes that are actually relevant for NASA in terms of contaminating other planet. Okay, so those things will be re-examined and quantified appropriately and compared with the NASA spore assay so that we will have a final compliance assessment and also risk quantitation. So what are the problems that we have right now? The technology gaps here: nucleic acid-free sampling device, nucleic acid-free reagents and consumable. I think I'm working with the JGI people, especially Tanya, to see whether we can come up with the appropriate database so that we can nullify this stuff from our sample. So mitigation strategies effective for low biomass, because these are all 10 picograms or low level. Can we get validated or reproducible data with this low biomass? And also one-click bioinformatics solutions, it may be tough, but hybrid risk models combining metagenomics and PCR data may be useful to measure and quantify. And this is another issue, dead and live evaluation. We are trying to do PMA-based analysis, but that may not be the method that can differentiate everything. There are some caveats there as well. And ultra-low bioburden wholesale reference standards has to be made so that we know how much we are losing, etc. So sample collection, sample processing and sequencing, bioinformatic analysis, and all these things needs to be charted out. And also you need to establish appropriate database for the relevant microorganisms and assess them. So one example that I'm going to show is, instead of swab sample, we are trying to collect more samples by using some aspirator-based, large sampling areas, which we call them SALSA, just a suction sampler. And then this itself, visually, you are seeing by swabbing, you are seeing very low, level of biomass compared with the SALSA of the same, 100 centimeters square. And then here you are going to give I'm putting a 16S qPCR analysis here. This is the clean room SALSA, that is this instrument, and this one is swab. So we have 1 or 2 log difference in terms of 16S copy number. Same thing in the microbiology lab is similar. And the level of contamination of the swab as well as this particular thing has to be improved a bit, but it is below 100 copies. One big issue here is if we are doing the relative abundance, we may not get the appropriate difference between the control versus our real sample, which is these 9 samples are real sample. As shower, which before getting into the clean room, we collect the samples, and then inside the clean room where the entrance level where they make a lot of cleaning, and far end side, which is not that much work going on, so basically you are not cleaning that great. So when you are seeing it, these samples that are far end not cleaned that much actually have considerable amount of sequences compared to the reagent-based control samples versus the clean room that is actually within this particular signal to noise ratio. So these are the things. What do we need to do with this? Are we going to discount this as clean samples, or by comparing the control, we need to see what are the things are left behind, even though it is low level of biomass. So that is another thing that we are going to do soon. So the recommendation next step is optimization of semi-quantitative metagenomics for low biomass and validation of the targeted digital PCR for quantitative assessment of spacecraft surfaces. And this will encourage NASA to continue regular workshops so that, this NGS is very dynamic. 3 years back is different, and 3 years from now may be different. But when we started doing this assessment, the 5-year term of a mission cannot change the technology. So we need to have international cooperation as well. So right now we are here. So system level testing, and then it may take 5 years for completing our thing so that the future mission can adopt DNA-based analysis. Still, we have not got the approval, but we are pushing for approval to get this system level testing of the DNA-based analysis, which we have done R&D based so far. So on other thing is I was telling is, with a few minutes that I will take, explain the omics approaches are predicted a lot of things, functional activities, and we need to validate them, right? So application of this research, one is Kalamiala story on clinical importance because human are going to be in there. And the other one is Klebsiella story, which is a kind of thing how you can eradicate some problematic fungi with the bio-based, antimicrobial compounds. Finally, how this, space-related microbes, when you're exposing it to space, can help us to get something back to the Mother Earth. So first of all, MAG, I don't need to explain what MAG is. So MAG is the one that we are continuously getting one set of things that come as unculturable. And then we actually went back to our cultured bacteria, which we thought it is not yet identified. And then we sequenced the whole genome of it. And then single cell genome was also able to come up with that. Then we named it as a Kalamiella pearsonii. Why I am telling this, when we published this one, this was not a pathogen. Our pathogen finder was based on the database, so it was not telling a pathogen. But as soon as we published, in India, they got this Kalamiella pearsonii as a uropathogen, and so it produces a kind of kidney stone. And then human disease caused by Kalamiella was reported in a New York hospital. And then where 56-year-old woman with the medical industry got this one. It was not a life-threatening, but it was 14 days of intravenous, antibiotics has to be, administered. And subsequently in Ireland, this particular pathogen was found as a causative agent for oral disease. A few days back, we got a call from Germany saying that in their medical hospital, they got this same thing from Kalamiella. So what I'm trying to say is these are considered as emerging pathogen, which we were able to get it with the MAGs, and then it's become a problem for the sustained living in space. So we had to make sure to eradicate them. The second story is the chemical biology, how you can use the one bacteria to eradicate the other one. So metabolic modeling of this International Space Station revealed key microbial interaction. What that means is Klebsiella pneumoniae is actually parasitic or ammensilism. That means ammensilism, they suppress the growth of other fungus, in this case, Aspergillus and Rhodotorula. So let me take an example of this. So we have got the Klebsiella, we got the Aspergillus from space station. We grow them together under various conditions. One is grow them as is under the normal gravity. So it is very nice, flower rate here. But when you grow them with Klebsiella, they actually act on those conidial spores and eating up. Now, if you are growing the Aspergillus fulminans under simulated microgravity, which actually not healthy as this one, that means it is related to the immunocompromised human there, right? So now it is completely bald if you're growing along with the Klebsiella. So this is actually a kind of model that predicted there, we were validated. So this particular Klebsiella is being worked on right now to develop an antifungal compound. The last story that I'm going to talk about is Bacillus sapphire 32, which one is we are exposing outside the space station. We took this spore and then kept outside for 18 months. Before that, we did all the whole genome sequencing and then did the background analysis of simulated Mars conditions, as well as whether it is resistant to various fungi, sporicidal activity. And this is a flight payload where you can simulate the space conditions or Mars condition on the space outside the space station. Okay, so in 2008, the STS crew took it out and then put it outside in this Columbus module here, and it it was exposed for 18 months and brought back to the ground and to analyze. So when you analyze, basically, most of them are died if it is exposed completely to UV, but one or two survived. So those survived things were actually, we did a lot of genomics. Genomically, there is no difference, but phenotypically, there are a lot of difference. One is the UV resistance, or for the control will die off in 2000 joules, but this one is completely not dying off even after 4000 joules. And the vegetative cells is same, around 200 joules. The vegetative cells of one of the space exposed vegetative cells of Bacillus pumilus die off, but 900 joules it is still growing. So the proteomics is showing that there is something, it is differentially expressed here. So now we actually took that metabolic differences to see what they are. And then these were actually become a product. That's what we call them as space to your face. So now it is being sold, and it is a age-defying serum. The main problem here is the main thing that is producing is hyaluronic acid. So these were mainly happened because of those data mining, omics data mining of these things. So that helps us to do. So what we want to do with JGI user group is chemical biology of this space exposed microbes that can withstand this microgravity and enhanced radiation to see why it is persistently living in the other planet. So with that, this is acknowledging all the people working along with me. So this is not a one-man job. It's a huge number of science team was working with me to help to get it done. Thank you so much. Sorry for overrunning.
Venkat, we have one quick question. We have time for one quick question. If you want to turn on your screen so we can see you.
Ah.
Oh, yeah, you stop sharing your slides. Okay,
Yeah.
great. All right. Do we have any questions from the audience here? Maybe we don't have a question. What's the next space? Oh, there's one. Yeah.
I was actually curious what the like procedure for the viability determination is. Is that also like a nucleic acid based approach, or like microscopy, or like some other other approach?
This PMA, which is a propium monoazide, which will intercalate the DNA. So when it is when the naked DNA and the compromised cells are there, this propium monoazide will go in and bind with them, so it will not be amplified with your PCR reaction. So the one that is not bind will be extracted DNA. So during the extraction, the PMA is degenerated. So the DNA from the viable cells are available for your downstream analysis. But as I said, one sample processing step is not going to solve all the problems, but because for example, spore and other hardy, coated microbes will not yield this PMA to get in.
Great, thank you, Venkat. And I think it's time for lunch now. Let's thank our speaker again for a great talk.
Thank you.